Hello and welcome. Here we will be discussing about the basics of plant tissue culture. So first of all, we have to think about what is plant tissue culture. Tissue culture is the culture and maintenance of plant cells, tissues or organs in a sterile, nutritionally synthetic media and environmentally controlled supporting condition, that is in vitro condition. In vitro culture, we can see that the culture is maintained in an artificial condition in tubes, glasses or plastics in a fully sterile conditions. The culture of plant seeds, organs, tissues, cells or protoplast can be done under a controlled and artificial environment. We can use plastic or glass vessels in aseptic techniques, and we can use a defined growth media. The growth and development of plant seeds and organs or any tissue under a controlled condition in an aseptic condition in a defined growth medium can be considered as in vitro culture. So what are the characteristic features of plant in vitro culture? We have to give environmental condition which in an optimal level, that is nutrition, light, temperature, everything has to be in optimum. So ability to give rise to callus as well as embryos, adventitious roots and shoots can be visible in plant in vitro culture. Ability to grow a single cell is also possible in vitro culture. That means we can culture protoplast, microspore or suspension cultures. And the plants are totipotent. That is the basic principle in plant tissue culture. That means in controlled condition, we can produce a fully regenerated plant from a small portion of tissue or explant. So what is totipotency? Basically, it can be defined as the potential or inherent capacity of a plant cell to develop into an entire plant if suitably stimulated. That means we have to provide the basic infrastructure for the development of the cell. It implies that all the information necessary for the growth and reproduction of the organism is contained within the cell. So just we are providing the environment, that's all. So one of the important term used in tissue culture is de-differentiation. That means capacity of mature cells to return to meristematic condition and development of a new growing point, followed by re-differentiation, which is the ability to reorganize into a new organ. That means organized cell can be converted into a meristematic condition, and this meristematic condition can be reverted back into a organic condition. So we have to think about the competency, the endogenous potential of the cells to develop in a particular way. The based on this competency, we can convert any tissue into a fully developed plant. So generally, we can say about something about the history of the plant tissue culture. So basic principle is a totipotency. And in 1938, Schleiden first introduced the basics of tissue culture, and Schwann in 1939 also did basics of plant tissue culture in plants and animals. Among the lower plants, any cell can be separated from the plant and continue to grow. Thus, the entire plant consists of cells which has the capacity for independent life. That is clearly demonstrated in almost all the plants. And Haberland in 1902 first aseptically culture. He made an attempt to culture plant cells in a nutrient medium or solution. And Gothard is the first one who did the first actual callus culture. And the most famous personalities in plant tissue culture can be considered as Murash and Scooge. They develop a very specific medium which still we are using for the plant tissue culture. And the important factors we have to consider while doing the plant tissue culture. First thing is the growth media. That means we have to provide all the nutrients for the developing cells. So that medium contain minerals, growth factors, carbon source, hormones. And also we have to consider about the environmental factors like temperature, light, photoperiod, sterility, growth media. Sterility is very important because we are providing a fully nutrient medium for the development. So naturally bacteria can or any other microorganisms can grow much faster than the plant cell. So we have to develop very strictly maintain the sterility of the medium. And the explant source. Usually the younger, less differentiated portion of the plant we have to extract. So that is known as explant. So we have to usually use younger explant for the betterment of the tissue culture. And different species show differences in its amenability to tissue culture. In many cases, different genotypes within a species will have various responses to tissue culture. Some will easily give somatic embryogenesis, but some cases it's not very easy to create somatic embryos. Somatic embryos means embryos developed from the somatic cells, not after the fertilization. So we can develop somatic embryos, but some species are very easy to generate somatic embryos. Even in some plants, we can see that they may not easily give callus culture also. We will see about what is exactly somatic embryos and callus. So this is the basic history and how we are doing the plant tissue culture. And apart from the hormonal, apart from the nutrient media, environmental factors, we have to consider about the hormones. Hormones are very important for the plant development. Two hormones or two divisions of hormones are actually affect the plant differentiation. One is auxin, which stimulate root development, and cytokinin, which usually stimulate shoot development. So generally the ratio of these two hormones can determine the plant development. That means if there is higher auxin to cytokinin ratio, there will be root development. If there is higher cytokinin auxin ratio, there will be shoot development. And if we are using almost auxin and cytokinin in equal amount, we can develop callus. That is the usual pattern we can see in the plant tissue culture. And this is the basic this table shows the important auxins and cytokinins. Considering the auxins, most importantly or commonly used auxins are IAA, indole 3-acetic acid, or indole 3-butyric acid. So many auxins can be used. Generally, auxins produce adventitious root formation. That is very important for the tissue culture and micropropagation, and it can also generate shoot formation, and also it will induce cell division, and also it will inhibit root elongation. Sometimes depends on the concentration of cytokinins. And cytokinins are commonly used. Cytokinins are kinetin and zeatin, which is a natural one, and benzyl aminopurine, we commonly used BAP or BA, then 3IP. These are the commonly used cytokinins, mainly for adventitious root formation and root formation, as well as they can promote cell division. So basic difference between the auxins and cytokinins is they can act their own, but after the combined effect, the result will be diverse. So for each plant, we can to identify clear-cut ratio of auxins and cytokinins for somatic embryogenesis, for multiple shooting or rooting or callusing. And another group of hormone are gibberellins, gibberellic acid. They mainly stimulate shoot elongation. And the other two groups, abscisic acid and also polyamines, can also be considered as hormones, which also influence the cell division and growth. So what is tissue culture? Basically, we can see the answer in this picture. And why tissue culture? We can discuss about that. So here we have a plant. We have to propagate it. Okay, we can take part a small portion of the cell. Maybe a piece of leaf can be cut and taken out in sterile condition. Otherwise, we can isolate the cells also. We can culture it in a nutrient media in sterile condition, and from that small portion of leaf we can produce large number of plantlets, and they can be grown later. It can be moved to nursery. So from a small portion or piece of leaf, we can produce large number of same plants. And we have to cut and take out small portion of the leaf as explant in sterile condition using, we can keep the plant steady using the sterile forceps, and here we can use sterile knife to cut the explant. And this explant has to be inoculated in a sterile either petal plate or tissue culture tubes or bottles, whatever we can use based on our need. And we have to inoculate this explant into the medium. From this medium, different, either we can induce somatic embryos or we can produce large number of plantlets. So why tissue culture? That means we can produce large number of plantlets from a small portion of plant. So there are different types of in vitro culture based on the explant we use. Based on the portion of the plant we use, there are different types of in vitro culture. There is culture of intact plant. That means we can culture seed, complete seed, or we can use seedling, or we can use embryo, or maybe you can use entire organ, or we can use callus. And there is suspension culture. That means cells are suspended in liquid media. And also we can use protoplast also in vitro culture technique. So first we can see what is seed culture. Seed culture means growing seed aseptically in vitro and artificial media. So this will increase the efficiency of germination of seeds. You may be knowing that some of the plants or seeds in some plants may not germinate. They have low germination capacity. In that cases, we need seed culture. And germination can be enhanced by application of plant growth regulators, and we can produce clean seedlings for meristem culture. So first of all, we can germinate the seeds in medium. Then from the sterile seedlings, we can use explants from the seedlings. That is also a possibility. And next is embryo culture. That means growing embryo aseptically in vitro condition in an artificial nutrient media. It is developed from usually embryo culture is needed for rescuing embryos. Sometimes we use a wide hybridization, wide crosses between entirely different species, and embryo can be produced. But usually embryo cannot germinate, or they cannot survive because of the hostile tissue around it. So in that case, we can isolate the embryo and we can culture it in a nutrient media. And thereby we can save that kind of hybrids. So there are some possibility of incompatibility between such different parents used for embryo production. So we can overcome that kind of incompatibility barriers. And also we can further develop in the production of plants. That means we can produce plants by non-sexual method. That means haploid plants can be produced. And we can also overcome the seed dormancy and also shortening of the breeding period. And about the production of haploid plants, we can say that ovules and ovary culture can be used. Okay, so usually the plants are diploid, but in some cases we may need to produce haploid plants for the production of diploid plants with similar genomic constitution. So a common explant for the initiation of somatic embryogenic cultures can be used, and we can overcome the abortion of embryos from the white hybrids, which usually happen at the very early stages of the development due to the incompatibility barriers. And also in vitro fertilization can be effectively used if we can culture ovary and ovule in synthetic media. And about the anther and microspore culture, this is a common technique which is used for production of haploid plants. Haploid plants may not be generated otherwise in nature. Very rarely we can produce haploid plants in nature, but in vitro system, we can develop haploid plants. Production of homozygous diploid lines can be produced through chromosome doubling. So it is very important for breeding technique. And we can also uncover if there is some mutation in the plant. It can be easily identified. And also we can use this culture system for in vitro pollination. Production of hybrids which are usually difficult to produce can be generated with the help of in vitro system. So in organ culture, any plant organ can be used as an explant. It may be shoot, then it can be called as shoot culture. If it is root, it can be called as root culture. And about the shoot apical meristem culture, that is the next system. Here we can use production of virus-free germplasm. Even if the plant is infected, we can generate virus-free plant from that infected plant. So that is, for that purpose we need meristem culture. And there is mass production of desirable genotypes using the meristem culture. And also we can facilitate exchange between different nations or localities. That means whatever plants we are producing in an in vitro system will be sterile. There won't be any microbes associated with. So it is easy for the in vitro plant generated plants for the exchange between different countries. There are so many laws regarding the cleanliness of the introduced material and exchange material. So that is easy to do in case of in vitro plants. And also cryopreservation. For cryopreservation means storing plants in a very low temperature in liquid nitrogen. So in case of in vitro regenerated plants, for the conservation of germplasm, we can use cryopreservation, and shoot apical meristem and meristem culture is effectively used for cryopreservation. And next term associated with in vitro culture is somatic embryogenesis. We can see that this is a cluster of cell. This is actually a callus. So first we can say, what is callus? Callus means a group of undifferentiated mass of cells. There is no differentiation here. It's only group of cell from a plant. So in the callus, we can produce somatic embryos. We can see the development of embryos here. These embryos later can be completely converted into a fully regenerated plant system. So from a group of callus, we can generate large number of plants. So it is a major path for regeneration of the plants, and also we can use this somatic embryogenesis for mass multiplication. We are developing embryos in a somatic cell system. That is why it is called as somatic embryogenesis. So it is a source material for embryogenic protoplast. From this we can isolate protoplast, which can be later utilized for protoplast culture. And also the same embryogenic somatic embryo can be utilized for large scale development of tissue and metabolites from the plants. That means we can use in bioreactors also. So about the organogenesis, usually either we can generate shoot or we can generate root in vitro system. So organogenesis may be shoot system or root system, and we can use this organogenesis method for mass multiplication and also the conservation of germplasm. We can produce a large number of shoots from a small piece of callus. This is callus. You can see callus here, and from this callus many tissues develop. Later a complete shoot will be developed, and followed by we can induce to produce a root also here. So we can produce many plants from a small piece of explant and callus. So this photograph showing micropropagated plants. In the test tubes you can see many shoots are developed. Here also we can see from the medium large number of plants are developed. So what is callus culture? Callus culture means it is just an unorganized mass of cell. A tissue can develop in response to injury. Even in natural condition, in some cases we can see tumor like growth in the leaves or sometimes the plant surface. So that is the tissue which usually grows at the time of injury. And same type of tissue development can be induced. That means we can induce the unorganized mass of cells by giving certain specific hormonal combinations, and we can multiply the cell again and again, so that we can produce large quantity of tissue, unorganized tissue. We can develop in either in solid media, or also we can develop in liquid medium. If we are using a liquid medium, it is called as suspension culture. I will show you about the media combination, how media is prepared after this. So in callus cultures, in some instances, it is necessary to grow the tissue to a callus phase before regeneration into plant, before organogenesis or before embryogenesis. The plant tissue has to be moved through a callus phase. That means unorganized or plant tissue cells in a unorganized state, that is callus. So for the generation of useful somaclonal variants as a source of different protoplast and suspension culture, we need a callus culture. And one of the important factor in callus culture is that we can use it for the production of metabolites. As you may be knowing that medicinal plants, most of the plants has medicinal properties. These medicinal properties are basically due to the presence of many molecules inside the plants. These plants can be produced directly from the callus or from the developed tissue inside a tube. We can produce the same metabolites as that of the plant system. So for the production of metabolites, callus culture is very essential. And also if we are culturing the callus for a long term, there is chances for changes in its chromosomal pattern or genetic constitution. So that means there will be variation in the plants developed from the callus. So such variations are called as somaclonal variants. These variations may be epigenetic, that means it can change, or there may be genetic factors involved. So using the somaclonal variations, we can develop many improved plants. That is one of the positive aspects of callus culture. In suspension culture, we are not using a solid medium. Here we are using only a liquid medium. So if you are using a liquid medium, the fact is that naturally if you are putting some cells or tissue or explant in the liquid medium, naturally it will go down inside the medium. So in that case, cells cannot multiply. They will not get enough air or oxygen. So in that cases, we can culture the tissue in liquid medium in a flask like this, and we have to put it in a not in a stationary condition. It has to be moved continuously. For that we have an apparatus, that is shaker. This apparatus, we can put the conical flask here with the medium and callus, and this equipment will be continuously rotating, so that medium inside the conical flask will be rotating. Along with that, the tissue inside that medium also will moving, and so that it will get enough oxygen. When the callus pieces are agitated or moved in a liquid medium, there is a tendency to break up. That means even if we are putting a large clump of tissue, by the movement, the cells will separate and we will get small pieces of callus or single cells. That kind of condition is known as cell suspension culture. That means cells suspended in a liquid medium. And one of the important factor in vitro culture is that we can create mutagenesis in vitro condition very easily. That means we can induce polyploidy, or we can introduce genetic variability. Naturally, there are chances for genetic variability. As I mentioned before, there is chances for somaclonal variation. That means changes in the genetic constitution due to continuous subculture, culturing again and again in nutrient medium. So along with that, if you are giving some compounds for polyploidy, we can generate polyploid plants inside the tissue system. And other important culture system is protoplast culture. It is very difficult to isolate and maintain a protoplast, but we can isolate and maintain and culture the protoplast, and from that protoplast we can produce callus, and then from that callus we can produce tissue culture regenerated plants also. So the living material, we can use plant cell. Even protoplast can be cultured from animal cell, and also bacterial cells can also be used. So if we are culturing different types of cells, even we can combine entirely different protoplast, or that means we can simply fuse an animal and plant cell inside the system. But fusion of animal and plant cell system may not be very practical. It cannot survive actually because they are entirely different. But entirely different species of plant cells or plant protoplast can be fused in a culture system, and we can produce very unique hybrids in the this type of culture system. So for that we need to identify the procedure or the media necessary for the protoplast culture of each plant. So combining protoplast culture, we can use this protoplast culture for isolation and culture, as well as we can fuse, as I mentioned before, we can fuse entirely different plant species in a in vitro system, and a unique hybrids can be produced. And in that way we can transfer the very unique characters like bacterial resistance or stress tolerance can be introduced in a different plant. It can also be used for transfer of cytoplasmic male sterility, organelle recombinance can be produced. So such a hybrid, that means fusion of somatic hybrid cells, we are not calling it as hybrids, but they are hybrids. That means first happening phenomenon is the fusion of cytoplasm, then only the nucleus will fuse. So the first formed one will be hybrids. Later this hybrids nucleus will be fused, and we will get a complete somatic hybrids. And another aspect, interesting aspect, is the in vitro flowering. That means in the culture system we can produce flowers. Beautiful flowers can be produced in a conical flask or tubes in our bottles. That means it has very important practical applications. It can also be considered as we can give as a gift for others with in vitro bouquet system. And how we can prepare the medium for the in vitro system? What are the contents and how it can be prepared and how it can be inoculated? That is the portion next we are dealing with. So the media used in plant tissue culture usually contain almost all the nutritional components. Then only the plant expand can survive. So in a nutritional component or that kind of nutritional composition is essential for growth and development of culture tissue. So the success of plant tissue culture depends very much on the type of the media we are using. And each plant tissue culture media contain the following essential components. That means it contain micronutrients as well as macronutrients, then iron, vitamins, carbon sources, organic nitrogen, plant growth regulators, and finally we use agar, which is only used as a gelling substance. It is not a nutritional component. Agar is used to make the liquid medium into a gel form. That's all. So we can consider each nutrient. First we have to consider the macronutrients. Why is it called macronutrients? Means they need in higher concentration, and it is present in millimolar quantities, and if you are giving more than 30 ppm, parts per million, or mg per liter, then it is macronutrient. And macronutrients provide both in anionic and cationic forms for the plant cells. So basically nitrogen, phosphorus, potassium, sulfur, magnesium, calcium are all can be considered as macronutrients. They have structural and functional importance, as well as they are important for protein synthesis, cell wall synthesis, enzyme machinery, membrane integrity. So for a lot of important functions will be done only with the help of macronutrients. Without macronutrients, plants cannot survive in vitro system. So if you are considering nitrogen, it is a major component in amino acids, and usually it is in the form of nitrate or ammonia. And potassium, it's an ion which is present in high concentration in the cytoplasm and also in the chloroplast. This potassium ions is essential for maintaining the ionic balance and also activation of many enzymes, a lot of functions, including the osmotic regulation. Then calcium. Calcium is an important cofactor for many enzymes, and also it is associated with cell wall and cell membrane integrity. It gives strength to the cell wall. It also helps the regulation of cell membrane structure. And about the phosphorus, it is very important for energy metabolism, and it is essential element in DNA and RNA. And magnesium, which is essential for enzymatic reactions and energy metabolism. Sulfur, which is important for the chlorophyll development. And considering the micronutrients, you might be knowing about the micronutrients. There are large number of micronutrients we need. Like that, plants also needs micronutrients. First of all, boron. That is the major component. Boron, manganese, zinc, molybdenum, copper, cobalt. All these are very important for the plant development. We are giving it in a less than 30 ppm concentration. And zinc, which has a very active role in protein synthesis. And manganese, very important for photosynthesis and enzymatic actions. Copper has also important function photosynthesis. Also, it is important electron transport chain in photosystems. And we are supplying the copper as copper sulfate. Molybdenum, it is essential for conversion of nitrate to ammonium, and we are supplying the molybdenum as sodium molybdate in the medium. And boron, which involves which is involved in many enzymatic activities, we are giving it as boric acid. Iron, which is an important enzyme cofactor, supplied in very low concentration, but it is supplemented with chelators, and otherwise iron cannot be absorbed. And we are supplied it as sodium iron EDTA, and otherwise iron deficiency can create hindrance to the development of plant cells. And along with the macro and micronutrients, we have to supply organic nutrients in the medium, which involves vitamins. And these organic nutrients are very important for the biochemical functions inside the cell. And cells can produce a small amount of vitamins inside it, but they require exogenous supply of different vitamins for the optimum growth. So most of the usable vitamins we are using is thiamine, pyridoxine, nicotinic acid, vitamin B complex, etc. Next is amino acid. The amino acids also sometimes we have to supply. Glycine is the most common amino acid we are giving, which is very important for nitrogen metabolism and plant regeneration. And considering the carbohydrate, we have to give the carbohydrate as for the carbon, and we are giving it as sucrose. Usually we are providing in the form of sucrose. And agar, as I already mentioned, agar is not a part of nutritional factor, but it is providing as a gelling agent, which is this agar is extracted from seaweeds. For example, red algae, gelidium, and gracilaria, they are also generally used for the extraction of agar. And also in some cases, we have to use some natural extracts like coconut milk, endosperm fluid of the coconut, or potato extract, yeast extract. That depends on the nature of tissue or plant we are using. Plant growth regulators, which are the most common for the development of organs or dedifferentiation or redifferentiation of tissue in the in vitro system. So plant hormone is a small organic molecule that elicits a physiological response at very low concentration And these regulators are important for the development of the different phenotypes. So it is like a messenger between the environment and genotype. It's providing the signal for the genome for what kind of development it needs next stage. So it acts as a messenger. So as I already mentioned, the hormones includes auxins, cytokinins, gibberellins, and abscisic acid. And different types of groups of hormones are this. And in the auxins, we have different types of auxins, IAA, IBA, like that. In cytokinin, which is used for promoting cell division and shoot proliferation, commonly used one as BAP and chitin. And gibberellins promote stem elongation and also bulb formation, maturation of embryo, like that. And abscisic acid, which can be mainly used for inhibition of shoot growth, and also it helps the germination of embryo. So different plant growth regulators has different function, and the combination of the growth regulators are actually controlling the type of development. And other important factor in the plant tissue culture made as a pH. pH has to be adjusted between 5 to 5.8 before the jelly. And for that, we may use alkali or acid to regulate the pH. And if the pH is below 5, the gel will not be formed properly. It will be very liquid-like manner. And the pH is above 6, the medium is very hard. In that condition, the plant material cannot absorb nutrients in a proper manner. So we have to regulate the pH between 5 and 5.8. So basically these are the different components we are giving in a plant tissue culture medium: macro, micro, organic, and others. Then we have to consider the pH. And finally, if we are going for solid culture or semi-solid culture, we have to give agar. So these are the basic components of plant tissue culture media. And how we can prepare a plant tissue culture medium. So nowadays we have synthetic sachet of medium. We can just powder is there. We can just dilute, or we can dissolve it in water, and we can directly produce a medium, sterilize it, pour it in the appropriate bottles or flask. We can directly use it. So otherwise, we have to weigh each and every nutrients and provide it into the liquid medium. So first, we are dissolving all the essential nutrients in double distilled water or deionized water. After the dissolving, for the dissolving, we may need to heat lightly the water. And after giving addition of all the nutrients, we have to warm it, and temperature has to be enhanced so that we have to dissolve agar. And for the agar, usually we give 8% agar. That means 8 grams of agar for 100 ml of the medium. If you are producing 1 liter of growth medium, we can use this 1 liter of growth medium in nearly 65 growing tubes. This is the culture tubes, the photograph. We can pour the liquid inside the tubes, and we have to keep it for some time. After that, the agar will make it a solid condition. So after pouring this medium, we have to cap it, we have to sterilize it, then only we can use it. So we have to boil the water containing all the dissolved nutrients, and we have to regulate the pH, then dissolve agar, and after this we have to pour the warm medium, because if it is cold it will get solidified. So we have to use warm medium, and we have to pour it into culture tubes nearly 4 centimeter height, or nearly 15 ml of media is needed for a single tube. And we have to place the tubes in a slightly slanting position, and we have to cap it in airtight manner. Then we have to sterilize in autoclave. Autoclave is a equipment for sterilization. Small mouse works like a pressure cooker, and for 20 minutes we have to put the medium and tubes in autoclave. It will be very sterile and it will be very hot. We have to take out the flask or tubes, and we have to keep in a slanting position and let it remain there for cooling. After some time it will get cool. The medium will get in a semi-solid condition, and this can be used for the inoculation. So this is the medium in prepared and it's poured inside the tube, and it was remained for cooling in a slanting position. So the medium is in a slanting nature so that we can give maximum surface area to the explant. So for the tissue culture, we put a small piece of explant in a sterile condition, and we have to cap it the tubes. We have to do it in a specially designed laminar airflow cabinet, which is a sterile cabinet. Inside this we are doing the explant inoculation, and for the preparation we have to wrap all the forceps, scalpel, towels, gloves, everything had to be sterilized. So everything has to be in sterile condition. We have to inoculate the piece of callus, and followed by, after some time we have to keep it. This is the leaf explant. After some time it will produce callus. Callus will start to produce from the sides. A group of cells will be developed from this. After some time it will be like this, a clump of callus, and from that callus we can produce multiple shooting. So this is the basics of tissue culture, and thank you.